Good morning and good day, everyone. So thank you for the warm-up session and the introduction. And today I'm going to talk about the application of sample multiplexing using in single-cell studies, and especially for the label-free multiplexing using the SNP genotyping. And the topics includes what strategies of the sample multiplexings are, the advantage of using sample multiplexing, and how to achieve it. And also, we will show some amazing testing results from our bioinformatics teams. And first of all, let me introduce the concept of multiplexing. So multiplexing is a method of combining multiple signals into one signal. And in the meantime, you still have a way to decode and demultiplex the signals into different ones. And so in biomedical industries, it is a widely used strategies to allow you to treat the multiple samples or cells or libraries, and they can be pulled together for a single experiment. And currently, we have a lot of applications in our biomedical industries. And first of all, multiplexing is a widely used technique for sequencing. And by giving every DNA sample a unique barcode or sequencing index, so the samples can be run in the same sequencing lane. And this optimized the sequencing resources as well as the capacity of the sequencing business. And after the sequencing is done, the data can be demultiplexed into different samples and sent to every customer. And for bulk RNA-seq, we usually treat the samples individually to have the individual libraries for RNA-seq. And Syncron uses the technique of the multiplexing to have a new product called Accurate Code. And for example, we have a 96 well plate, so we can treat with 96 samples on the plate at the same time. And what we do is to give every sample in every well a unique barcode. And after that, the samples, the cDNA libraries of different samples can be pulled together for later procedures and for sequencing. And this is going to make the bulk RNA-seq more cost efficient. And by the way, we are not only have the 96 well plate, but also have the 384 one. And also, this is not only for bulk RNA-seq, and the multiplexing is also widely used in single-cell studies. And to achieve the high throughput, what we usually do is to give every cell a unique cell barcode. Here you can see from the bead. So basically, we have a large number of microbeads, and by using our JAXScope technology, we're going to load the cells from the cell suspensions onto a chip, and then we'll also load the microbeads with different cell barcodes onto the chip to make sure that in every microwell there is one cell and one beads with only the unique cell barcode on that. And after that, the mRNAs captured by the poly-DT tail can be pulled together for a combined library. And later, after we got the sequencing data, we can demultiplex the cells, the RNAs from the library into different cells based on the cell barcodes here. And this is basically how the high throughput single cell technologies are achieved. And I think this is also part of the reason why the single cell technologies are developed very rapidly. And based on that, we also modified the microbeads to achieve other types of single cell multi-omics. And so if you are interested in doing like the TCR, BCR, or the SNV information or dynamics of the RNAs, so you can also use the modified beads for single cell multi-omics with us. So if you are interested, please contact us. Our marketing team will give you more information. And next, we are coming to the sample multiplexing in single-cell technologies. So here, the sample multiplexing means that the single-cell solutions from multiple sample origins are put together to build a single library. And we have several strategies for that, and we'll introduce the strategies in the following section. And here are the advantages of applying sample multiplexing. The first of all, and the most obvious one, is and it is very cost efficient, especially when you treat it with a large number of samples. This is because after pulling the samples all together, many of the steps can be done for only once, and saves the time and the money. And besides, the multiplexing can also minimize the battery effect caused by technical differences. And even though we have a lot of battery effect removal algorithms available to help us remove the technical difference, it would always be great if we can minimize it at the very beginning in the wet lab. By applying sample multiplexing, so the sample treated all together using exactly the same reactions and procedures at the same time, and also with very similar sequencing depths. And this minimizes the battery effects caused by the experiment. And besides, sample multiplexing helps to identify multiplets. So doublets or multiplets are those artificial signals generated from two or more cells during the experiment. And this is a very common phenomenon in single cell studies, and it will make some confusion, especially for the study of cell differentiation. Here I showed a figure of the effect of doublets when building a cell trajectory. You can see that the doublets are at the round of the trajectory to make some noise. And when we usually, if the sample multiplexing is not applied, there are basically two methods to identify the doublets. One is by using doublet remover softwares, and the other is to identify it through the marker gene expression patterns. So if a cluster of cells express the marker genes of more than 2 cell types, it is very likely that the cluster of cells are doublets. And here, after we used the sample multiplexing, we'll find that a big fraction of those multiplets are those data points from cells from 2 or more samples. So this can be recognized and demultiplexed and removed when we are doing the sample demultiplexing. And there are mainly two strategies of sample multiplexing. The first one is barcode-based sample multiplexing. And by giving every sample a unique barcode, so we can achieve this barcode-based sample multiplexing. And usually, basically, what we do is to use antibodies or chemistries to link to the cells of the samples. And so our company have a barcode-based product called CleanDex. And the CleanDex uses the click chemistry for efficient and unbiased sample tagging. And so after that, after the chemistry is linked to the samples, so these samples can be pulled together as a single library for the later procedures. And the good way of using the chemistry is that so when you're using the antibody-based labeling, it is very relied on the specific protein markers. But when you use the chemistries, it can target a wide range of surface marker proteins across different species, and this enables a wider range of applications. So after sequencing, the cells from different samples with different sample tags can be demultiplexed based on the sample tags. And however, applying this technology, will cause extra steps and extra time for sample tagging, and sometimes the quality of the samples might affect the successful rate of the sample tagging, and it increases the cost because of the sample tagging procedures. And also, it requires greater sequencing depth, as now for every RNA, besides the cell barcodes and UMIs, we also have the sample tags on that. So we need to sequence a little bit more. And here we introduce the other strategy for sample multiplexing, which is the label-free SNP-based sample multiplexing. And this strategy is based on the SNP variants in different samples. So the most important thing here is to make sure that the samples are genetically distinct. And so the samples from the same donors could not be multiplexed into one experiment. And this strategy is especially suitable for those clinical samples who already have the genotyping data like WES available. And as long as these samples have enough genetic variants, they can be mixed at a very straightforward way and without any sample tagging procedures. And so the experiment of this sample tag of this multiplexing is very easy, and it can be treated with as the same with treating with one sample. And so after that, when you want to demultiplex the cells into different samples, and this relies a lot on the bioinformatics tools. It can help you with the SNP information to assign the cells into different sample origins. So in the next section, I'm going to introduce the softwares we use to do the sample demultiplexing for single cell data. So the recommended software is called Demultiplexing. So it is a software that can be used to decode sample identities and also identify the multiplets when multiple samples from genetically distinct samples are pulled together for single cell sequencing. So there are two parts of the inputs. The first part is a single cell BAM file, which is a standard output file from the pipeline. For example, if you use Telescope to run our company's single cell RNA-seq data, you would have this file as the output, and this is also the same with Syringa. And in the other part is the genotyping data, and we recommend to use WES. So after you calling SNP with this WES data, you are going to have the VCF or BCF file containing the SNP information of every individual. So as long as you have these files, you can start to analyze with Demultiplexing. So Demultiplexing will call SNP for every single cell using the BAM file, and to compare with the reference genome data of the WES. And so so it will calculate a probability of the cells to each of the donor and find the most likely donor of that cell based on a maximum likelihood framework. And in the meanwhile, the doublets will be identified and removed during the demultiplexing. And also, there are some other softwares available, like for example, the Viral. This is very similar to Demultiplexing, which will also use the genotyping data for demultiplexing. And also, there is one called Supercell. So by using this software, you don't have to use the genotyping data. This software will simply clustering the cells into different cluster of regions based on the SNP called by single cell. So in the later part, we'll also compare Demultiplexing with these two softwares. Here, let me show you some testing results from our bioinformatics teams. So to evaluate the SNP-based demultiplexing accuracy, we combined 5 of the single-cell RNA-seq data samples into one virtual pooled library. And after that, the demultiplexer will be used to recover and assign cells into different sample identities. And we use the true positive rate to evaluate the accuracy of the software. And here, the WES data is used as a genotyping data reference. And you can see from the testing result that most of the cells are assigned back to the right sample origin with the true positive rate from 97% to 99%, which is very high. And also, because this is a virtually pooled library, there should be no multiplets for the multiplets from different samples. So let's say if the percentage of the multiplets are 5%, and we're counting for this multiplet effect, and the accuracy is still over than 90%. And from the table, we can see that the number of SNP loci in the WES data are all very high. And we are wondering the requirements of the WES data that can be used for sample demultiplexing. So we did some further testing on the impact of the number of SNPs loci. To test with the impact of number of SNP loci, we randomly downsampled the WES data into different number of SNPs. And from the results in the table, we can see that when the number of SNP reaches 40,000, this accuracy is already very high. And when we increase the number of SNPs more, the accuracies are not going to change a lot. However, when we have very limited number of SNPs, like we say here, we have 10,000 number of SNPs in this data, and we found that the accuracies are limited. So we think that it's very important to include enough SNP information. And this is very similar to the gene expression in single-cell RNA-seq data. Let's say that when we have very limited number of genes expressed in the cell, it would be very hard for us to annotate the cell types of the clusters in the single-cell RNA-seq data. So for SNPs information, it's always the same. And so from these testing results, we recommend to have at least 40,000 SNP loci, and so meaning that for every sample's WES sequencing data, you should at least have 2G sequencing data. And we also tested with different types of genotyping data as a reference for the single-cell RNA-seq demultiplexing. And except for WES, we also tested with SNP code by bulk RNA-seq, because it's a very commonly used technology. And the simulated data set is combined of 3 scRNA-seq data with the paired bulk RNA data available. And demultiplexing is used for the testing. And we found that the number of variants here in every sample is all very limited, and so also leading to the accuracy from 60% to 40%. And so we can see that the sample with more variants with this S6 sample have higher accuracy of the demultiplexing, which means that the number of SNPs are very important for demultiplexing, and also the bulk RNA-seq are not sufficient to provide enough information for us to demultiplex the single cells. And as for the WGS, that's totally fine, as I think it can provide even more information compared to WES. However, because a large fraction of the reads in single cell data are mapped to the exon, so we think that WES should be the best choice to work with the genotyping reference. So in the last part, we tested with the softwares. So for the same simulated data combined of three RNA-seq data, we tried both demultiplexing and viral because these two need the genotyping data as a reference. We found that for all of the three samples, demultiplexing have higher accuracy compared to viral. So we think that compared to viral, we will use demultiplexing as a recommended software. And there is also a software called Supercell, which by using this software, you don't have to use the genotyping data. And as shown here, this software is going to cluster the cells into different clusters, and this is based on the SNP code by every single cell. So the cluster could come from a single donor, but the disadvantages of using this software is that you are not going to know which cluster is from which donor. So unless you have the genotyping data, you are not going to know the sample identity. But still, it has some applications. For example, here, this is a data set mixed of the maternal and fetal cells. So first, by using the supercell, it is going to cluster the cells into different clusters. And then with the help of the gene expression or the cell type markers, we are going to know which cell clusters are fetal and which are maternal, because the cell types and gene expression patterns of some specific gene would be very different in fetal and maternal cells. But still, because we want to know which cells are from which sample, we still recommend to use the genotyping data by using the demultiplexing for demultiplexing. So at the end, let me summarize what we have today. We talked about the sample multiplexing strategies used for single cell tech studies, and there are two main strategies. One is the barcode-based sample multiplexing, and one is the SNP-based sample multiplexing. And the SNP-based sample multiplexing can pull the genetically distinct samples to build a single library, and the demultiplexing process can be achieved using demultiplexing with accuracy over 90%. And WES is recommended as the genotyping reference data, and we should at least have 40,000 SNPs and at least 2G sequencing data for every sample. And when you apply in SNP-based sample multiplexings, samples from the same donor should not be pulled into one experiment. And sometimes when we treated with some cell types like red blood cells with very little RNA content, this will have very little information from the cells, and these cell types may end up with lower demultiplexing rate. And that's all for today's topics. And thank you so much for attending today's webinar. And if you have any questions, or if you are interested in doing sample multiplexing, you can contact us through the email info@synchrone.bio, and also you can contact us on Synchrone Bio homepage through the contact us form. And here comes the Q&A section.
If I have multiple collections from the same patient at different time points, how can I achieve SNP-based sample multiplexing?
Okay, so then I think the experimental design would be very important. And let's say when you are doing the project, you should have biological replicants. And if the biological replicants are available, we can still achieve the SNP-based sample multiplexing. So for example, say you have two time points, one is treatment naive and one is after treatment. And you have, for example, you have 4 donors, and I would recommend you to do like two combined libraries, one and for each combined multiplexing, you should have 4 samples from different donors. And in the meanwhile, I would suggest you to include this 2 samples from the treatment naive group and 2 samples from the after treatment group to also minimize the battery effects between the groups. And if you have more samples, that would be more complicated for this experimental design. But as soon as you don't include the samples from the same donor into a single experiment, that would be fine.
Another question coming in is, how many samples are recommended for multiplexing in a single experiment?
Okay. So this is related to the cell loading capacity of the experiment. Say, let's take an example as our JAXscope technology. So usually the normal chip we use, we have a expected cell number of 10,000 cells. And when you multiplex like 3 samples into one experiment, you should expect to have like 2,000 to 3,000 cells per sample. That's still acceptable. And in the meantime, we also have a HD format chip. And by using the HD format chip, you will end up in getting around 30,000 cells. I think this is very suitable for multiplexing. And so by using HD format chip, you can kind of multiplex for 5 to 6 samples. You still have like around 5,000 the cells per sample. I think that's enough for the study of single cells.
Thank you. Have a nice day and see you on the next webinar. Bye.
Thank you so much for attending.